at the end, which will be being collected through our YouTube broadcast. So for us, it's all about collaboration. You know, I won't spend much time talking. One of the things we realize, and probably you can share some of your experience in your countries, that many of the solutions that we have put in place here have been through extensive collaboration, intensive collaboration between, you know, startups, mid-sized companies, multinationals, healthcare institutions, hospitals, research institutes, universities, from many departments, not only life science, but, you know, engineering, computer science, and so forth. So if one good comes out of this pandemic situation, hopefully it'll be a more collaborative world. And from now on, you know, it's about working together, because we all need to get out of this together. There's no one person or one country coming out of this together. So again, welcome. Without, you know, much delay, I think I'd like to invite you all to present. As you know, there'll be, you know, 10-minute presentation, and then at the end we're going to have the questions. So, you know, as for the solutions, we're going to have solutions, you know, presented here, you know, that related to diagnostics, to, you know, supporting patients at home, to helping organizations go back to work, to organize, you know, life in the new environment. So it's very wide range. In a way, this pre-event with all these startups, it covers more or less the same topics that are covering in the full event. It's just, you know, very, very focused. So my understanding now, we're going to start with Gustavo. You're going to help me here. It's going to be the first one. It will be first company to be presenting will be
the first one is going to be NanoScent.
NanoScent, yes, very much. Sorry, thank you for this. So NanoScent is a startup from Israel. They've been playing a long time for in the field of, you know, lung cancer, as far as I know. They understand patterns of metabolites, and they have applied all their knowledge to fight COVID. So without much further ado, I'd like to welcome NanoScent to present. Please join us, and I will turn off my video camera now.
Thank you very much. First, let me say thank you to Gustavo and Albert Einstein Hospital for this session. What a great idea for collaboration worldwide. Thank you for that. I will share my screen now. You can all see my screen, right?
Yes, we are. Yes, we can.
Okay, thank you. So as presented before, my name is Nitzan Givon. I'm a research associate at NanoScent. We are an Israeli startup company. The company has been developed a sensor for detecting VOCs. This been going on for about 3 years right now. Our company, it's not something that was established to deal with COVID-19. However, our technology, which is again very much established, we can now provide it in order to detect COVID-19 patients. We are looking mainly to find the people with COVID-19 in a way of mass screening. NanoScent, with the rest of the world, figured out that there is a problem with the current methodologies to test people with COVID-19. These methodologies, they take time, and they have some range of accuracy to them. Excuse me, and there is a massive problem for testing a lot of people. And what we saw with our technology is that we can apply the technology in mass screening environment. in order to find those people with COVID-19, and as importantly, to find people that don't have COVID-19. And the idea is for those people who are healthy, we want them to carry on with their ordinary life. Their life should not be stopped because of COVID-19, as I'm sure we are all experiencing the same thing. I know in Israel, people
Nitzan, sorry for interrupting, but we cannot see your screen. Could you please share it again?
Yes, of course. What happened? Do you see it now?
Yes, now I can see.
Great. You. Okay. So, NanoScent technology is offering rapid tests for screening of COVID-19 in mainly public places, which means places like stadiums or places like airports, universities, schools, whatever place that requires a gathering of a lot of people. Our technology is here to provide mass screening. And the test itself is very quick. It takes about 1.5 minutes from start to finish, which means from the point that I take the sample to the point that I get an answer, whether I'm healthy or whether I'm suspect for having COVID-19, it's 90 seconds. And this is something we're still working on lowering this time of response. So as I mentioned before, the test is very quick. What we do is we test the nasal air. The subject, in order to test himself for COVID-19, has to blow into an air trap. This air trap is being connected to our device. Our device takes the sample in, and our sensor analyzes the sample. The sample itself, these are the VOCs that come out of our nasal air, and this is what our sensor can detect. It is well known that you get a different pattern of VOCs for healthy people and people that has COVID-19, as well as other diseases too. So we applied this knowledge and our technology for the testing of COVID-19. So basically, the way our technology works is that you really get the answer on the spot. You don't have to go to a doctor, and you don't have to take the test to a lab. You really get it, the answer in 1.5 minutes on your cell phone. And as I mentioned before, this is some of the use cases for our technology to work with. And I can show you a little short demonstration of how our device work, if you will. But before that, I will tell you that we have been conducting clinical trials in Israel, and now we are trying to conduct clinical trials around the world. Our first and initial results show an accuracy of 90% in detection of COVID-19 patients. So if you will, I'll show you a short demonstration of how it works. I think I have to stop the sharing. So very easily, this is the air trap. This is how it looks. I'm going to take a breath through my nose and hold it for 5 seconds and blow into the air trap. Once the air trap is filled of my sample, which basically contains the different VOCs, I can attach it to our device. It's a very small device. You can hold it on the palm of your hand, and you have to put it to the device. Sorry, I forgot to put this part. You just put it, and with your cell phone, with an app that we have developed, I can activate the device and get the result. You can hear now the device taking the sample in, and the analysis begins. And hopefully I won't be positive for COVID-19. It's very important. And see, I got an answer that I am COVID-free. That's good for me. And this way we can mass screening many people in parallel in large places that needs a gathering of a lot of people together. This is it.
Thank you very much, Nitzan. This is an amazing prospect for all of us, for humanity, and hopefully for your company as well. We'll certainly talk to you about clinical trials. Next week we have an academic research office here. We're very eager to test new solutions, and we're doing this all the time. So congratulations.
Thank you.
So,
good luck.
Yep. Hello. So very good. Now we're gonna travel to Europe, to Germany actually. We're gonna have Altoida, which is a company which was developed to support patients with Alzheimer's. But as we all know, this pandemic is presenting a really hard burden on individuals, particularly old people and their families. I can speak to my own family, which has its own challenges right now, like many other families. And I think, I'm going to have Irene Meyer, who's also an innovation director, presenting the solution and how they're playing a role within this pandemic environment. Irene, so please come to the stage, and I'll fade into obscurity here.
Perfect. Thank you very much. Can you see my screen?
Yes.
Okay, fantastic. Well, thank you very much for the invitation to speak here. It's an honor for us. And as you introduced, we are a company working with Alzheimer's disease, and more specifically digital biomarkers to detect Alzheimer's disease or the risk of developing Alzheimer's disease. So the way we do this, I'm going to quickly introduce how the technology works, but also what we've been able to do in adapting to the current situation. So Alzheimer's disease is the most common form of dementia. It happens in about 1/3 of the people over the age of 80. And there's about 500,000 to 1 million cases in Brazil alone. And the way we work, one of the big problems is it's not easy to diagnose early when intervention would be most effective. And so we've come up with a tool where we work with active digital biomarkers. So this is an application on a smartphone or an iPad. And we start with motor tests. So we have the participant do a series of tests, following the circle, for example, this squiggly line, and then here's more of a reaction time. As you can see on the bottom, we measure motor speed, accuracy, we look at the eye tracking, because this is shown to be a very sensitive measurement also for cognition and cognitive decline. And then we have something that's like sets us apart, which is an augmented reality test. So what we do is we have subject hide 3 virtual objects in an actual room. and find them again. And that would translate to, for example, losing one's keys at home and then finding them again. And what we measure is every possible input during this test. So this includes gait of the patient walking to place the object, the balance, we call this visual spatial memory to remember where you placed something, and then also finding it again, orientation, planning. In short, this is a 10-minute test in total, and we measure over 700 individual features. The second bit of this test is also an augmented reality test, where we also have participants hide 3 objects in a room, virtual objects, and find them again. But the difference here is that we give them a pre-specified order that they'll have to remember, and we call it perspective memory, and that would translate to you remembering your shopping list, for example, when you go to the supermarket. And we have a second aspect here as well, which is it's a dual task. We distract the patients a little bit, and that's what really happens in real life, right? It's not just like ever one thing. And then the phone rings and you get an email and you forget like what we were supposed to do in the first place. So we measure all these aspects. And then also with the pandemic, very important, and this was mentioned briefly before, there's a lot of people who become lonely due to isolation. The increase of depression is marked due to social distancing, due to staying at home, quarantining. And so we introduced a voice test, and we're not interested in the grammar of the participant, the patient, but we measure the agitation, depression, irritation, and that's also a big contributing factor to Alzheimer's disease. And we actually expect this to increase during this pandemic and after. And then this test also has a second part, which is where we ask the patient to look at an image. Then the image will disappear, get blurry, and they'll have to recall this from memory. So what we do compared to conventional measures, which is a lot of paper pencil tests that are based on speed and accuracy, and usually it measures one, you have one question and get one response to it. And what we do is we use these smartphone sensors, the accelerometers, gyroscope, magnetoscope, the camera as well for the eye tracking, the microphone for the voice, and the touch screen to test how patients interact with the device. And we are able to measure over 700 features with that. They will feed into 11 cognitive and behavioral domains that we typically know from like a neuropsychological assessment or neurological assessment. And then what we did is, and here these things are the most important ones, we did a study to validate our tool in about 580 subjects between the age of 55 and 99 years of age that were divided into healthy controls, mild cognitive impairment, which could be described as the pre-stage of dementia and Alzheimer's disease, and then Alzheimer's patients. And we had a ground truth that included neuropsychological assessment, Alzheimer's biomarkers as well, so amyloid and tau CSF, a brain MRI, an EEG, and a genetic risk gene, ApoE, whether patients have that or not. And we tried this, or we tested this with, at the time, 320 features. Today, we increased to over 700. And we basically showed that we're able to differentiate healthy controls from people with MCI due to Alzheimer's disease 6 to 10 years prior to symptom onset, which typically is not possible. Alzheimer's is usually diagnosed once the patient starts noticing symptoms like forgetfulness, and attentional problems, but also irritation. What we can do is we have a 94% accuracy rate to distinguish healthy controls from MCI due to Alzheimer's disease at these very early stages. But we also realized that we're able, with 91% accuracy rate, we're able to predict who is amyloid positive versus negative. So amyloid is one of the hallmark proteins of Alzheimer's disease and pathologies. And if we have ApoE4 status, so the genetic variant that it can be determined with a blood test, we get to 98% accuracy rate. So this is to distinguish amyloid positive versus amyloid negative MCI patients when we have the ApoE status. What this means is, and how we compare to our competitors as well, is a lot of people assess usually just one domain. They either just look at eye tracking or at motor test or planning, for example, and we really include everything into one assessment that is only 10 minutes long. And we also had participants do the test in the clinic and then take the test home. And this, I think, is where the importance comes in for a pandemic as we currently live it, and social distancing, and ensuring that elderly people still can get the monitoring. They can still be tested and still followed, and they can do it safely from their home environment. Again, this is our accuracy rate for diagnosis. What we can also do is monitoring patients long time. And we have a very, very sensitive tool, so there's very little learning effect from one test to the other. It actually allows for repeated testing and gives us very accurate and sensitive measures. And what's most important here, when we briefly talked about regulatory aspects before, we are FDA cleared, EMA cleared, and also Anvisa approved since last October. We also have a strong partner here, or a unique partner, distributing partner, BioCare. And our efforts during the pandemic was really to come up with this smartphone version, because a lot of people don't have a tablet at home, especially in Brazil, for example, where they can do this test. So we developed for iPhones and Android. It works better on iPhone at this point, and this is still a process ongoing. But we have a lot of study. This is like 12 years of research. We have multiple studies. The one that I showed as well just before about the 94% accuracy rate for diagnosis was just accepted for publication, so should be online in about 2 to 3 weeks. And the testing from home, we believe, is important and crucial. And there's also reasons to believe that actually COVID itself will have an impact on cognition and cognitive decline. So thank you very much. I look forward to your questions.
Thank you so much, Irene. This is really amazing. I know there's some questions popping up already on my WhatsApp. We'll put all together at the end of the presentations. And, you know, from my own family and experience, I think this is going to be a great solution for people being rightly diagnostic during this pandemic, so we can deal with this more effectively. So anyways, I think we're moving along. You know, it's a good time to be here. Now we're gonna go cross the Atlantic again. I'm gonna go to Canada to hear from fellow Canadians. Hi, how are you? I'm not sure what you know, Dr. Scott, but I'm also Canadian. So there you go.
Oh, excellent.
So, yeah, I lived for a long time in Toronto, but moved back to Brazil. So welcome to our show. You know, it's hopefully something will come out of this. We have a very inquisitive audience already. I'm very eager myself to hear about what you're doing in Saskatchewan. Thank you very much. I'm fading out now.
Excellent. Well, thank you so much for the invitation to participate in this event. I mean, this is absolutely amazing to see all these people from around the world coming with these unique technologies and perspectives as we try to deal with this unprecedented challenge. As mentioned, my name is Dr. Scott Napper. I'm the Acting Director of Research at VETO Inovac. And what I'd like to do is to use my time this morning to first tell you a little bit about our organization, then to describe to you some of the work we've been doing with COVID-19, things that might be of interest and application to others, and finally to describe our efforts in terms of developing a vaccine for COVID-19. So VETO Inovac, we are located within Canada, within the city of Saskatoon. We are a proud member of the University of Saskatchewan. Our organization has been around for about 45 years now. The first 30 years of that was largely spent on livestock infectious diseases. And then about 15 years ago, we transitioned looking more into pathogens of consequences to human health. But those origins are really important to us in terms of developing the infrastructure and expertise of working with these animals, and in particular these large animal models of human disease. We're quite a large organization. We're up to about 170 people now with an annual budget of about $20 million a year. Our priority is to do high quality research that has real world impact. So even though we're part of the university, our driving force here is to come up with technologies that will go into real world applications. That's well demonstrated. We have 10 commercialized vaccines so far, over 150 patents. So very aggressive in terms of translating our science into real world applications. We are home of Inovac, which is Canada's largest containment lab. So this is the largest investment that the Canadian government has ever made in science infrastructure. It's one of Canada's 10 major science initiatives. This thing is absolutely massive. It's about the size of a football field. We have 10 animal rooms. At any given time, we can have over 1,000 pigs in level 3 containment. So when you start to think of what that would translate to in terms of ferrets, hamsters, or mice, virtually no limitations in terms of space. So absolutely astounding state-of-the-art facilities. We recently received funding from the federal government to also build a vaccine manufacturing facility within Inovac. So we're starting construction on that this summer. It should be done within the next 18 months. And that's going to be really important because that's going to allow us to go from the first idea and doing basic research all the way to producing a vaccine that's ready to go into clinical trials in human beings. So tremendously exciting. And again, this idea of our experience and our ability of our animal handling team to work with different species. So we've had everything from bats to bison within Inovac. So a lot of capabilities there. And again, the idea of all of these different animal models we can work with for these different diseases. So a lot of these are of interest for the animal health sake itself, but in other cases, it's using these large animal models as more effective models of human disease in terms of developing vaccines and therapeutics. So we've been leading Canada's response to COVID-19. That very early on, we sort of had an inkling of how bad this may get. So we were very aggressive in responding to this. And we're very proud to say we were the first lab in Canada to isolate the virus. We were the first lab in Canada to establish an animal model. And again, those animal models are so critical that if you want to be able to test vaccines and therapeutics, you have to have a good model in place. And we were the first in Canada to have a vaccine that was under development. So all of this was happening as early as January. We were starting in this process. One of our big priorities as a major science initiative is not just in terms of our own research, but enabling the research of others that we appreciate as we have this unique infrastructure, as we have this unique expertise. It's not just about advancing our own research, but it's helping people from around the world. And one of the things we're tremendously proud of over the past few months, we've been working with dozens and dozens of different groups as we're testing their vaccines and their therapeutics. And that's both within our animal models, but we also have the capabilities to do in vitro screenings. So to test these molecules in tissue culture, to see if they have potential as antivirals, and then we can then translate that looking at animal models. So a big part of what we're doing is making sure we're helping everybody come up with an effective treatment for this. So the two animal models we've been working with are within the ferrets and the hamsters. So for both of these, we're looking at an intranasal challenge model. So the virus, as you can see, goes directly into the nose of the ferret or the hamster. We have different doses that we can give. So with ferrets, we initially looked at both a high and a low dose using the strain of COVID-19 or of SARS-CoV-2 that we isolated right here at VETO. With hamsters, looking at a slightly lower dose based upon their smaller size. And so what we're seeing when we look within the ferret model, it seems to be quite a good animal model, and not surprising that that was discovered for both MERS as well as SARS, that in terms of these respiratory infections, the ferret's quite effective. So we're able to see the viral RNA within the nasal and oral swabs. It spans about a 10-day period before they clear the infection, with a peak happening somewhere between days 2 to 6. We can also isolate the virus from the nasal washes as well as from the upper respiratory system. So again, the idea that the virus is replicating within these animals, making it an effective model. What we see within the ferrets with respect to clinical signs, that they don't seem to get particularly sick, that if we look in terms of behavior, in terms of weight loss, in terms of temperature, not a whole lot of clinical signs. So a fairly mild form of infections. When we look within lung pathology, what we're seeing is pathology that's consistent with pneumonia. So again, the idea that there is lung damage occurring here. So what this does, this provides a number of different measures for us to be able to evaluate the efficacy of different vaccines or therapeutics, both in terms of their impact, in terms of the amount of virus that's present, as well as minimizing the damage that might occur within the lungs. What we see within the hamster is a more aggressive form of infection. So when we look at that span at which we can detect the viral RNA, it's a shorter period and it peaks a bit earlier. Perhaps the biggest difference is in terms of the clinical signs, that within the hamsters, we do see quite a bit of weight loss, losing up to 20% of their weight. We're not seeing a lot of indications in terms of temperature change. With the ferrets, we were seeing that all of the animals would eventually clear the infection on their own, whereas within the hamsters, about 10% of the animals, that this will represent a fatal infection. And as you'd expect with that, we're seeing greater pathology within the lungs of the hamsters. But again, within both of these animal models, we've been able to use them to show the efficacy of different treatments. Now, in terms of our efforts to develop a vaccine for COVID-19, so what we're focusing on is making a subunit vaccine. So that means we're taking a piece of the protein from the virus and using that as the vaccine target or the antigen. And so we're looking at a couple of different antigens. So we have the spike protein, which is going to be exposed on the surface of the virus, as well as specific subunits within the spike protein, like the receptor binding domain. So for all of these, it's really important that you're able to produce large and reliable quantities of the antigen, and that it's going to be adopting a conformation that mimics what it looks like as it's present on the surface of the virus. So we've taken great care that with the spike protein, we know it's meant to form these trimer structures, making sure that's what we're seeing. Another critical part of developing a vaccine is not just having the appropriate target, but you also have to have the right adjuvants in place. So this is what's going to be promoting the immune response. And it's not just in terms of the magnitude, but it's also the type of the immune response. So one of the things we had developed here at VETO was a proprietary adjuvant based upon 3 different components that stimulate the immune system in a different way. And what we knew about this adjuvant is that it tended to promote what's called a Th1 response, which is typically the type of response you would want to see for a vaccine such as this. It tends to induce very strong responses. Sometimes even with a single immunization, you can get a very strong response. We also know that it works very well with proteins like this that are heavily glycosylated. So there's a lot of sugars attached to the proteins. And we've been able to show for other respiratory infections like COVID-19 that this has been very effective. So I'm just going to show you some sort of high-level results of 2 of the vaccines we've been developing. So what we're looking at here are the neutralizing titers. So this is in the production of antibodies that are able to neutralize the ability of the virus to cause infection. So looking at the 2 time points corresponding to the initial immunization and then with a boost, you can see that both of these vaccines induce very high neutralizing titers, quite consistent responses as well. And even when we compare this to animals who have been challenged with the virus, we're seeing stronger neutralizing titers with our vaccine than you would see in response to challenge. So basically, this is going to give you better protection than a person who was just infected and managed to clear the infection. We're also seeing that our vaccines are able to protect animals from challenge with the SARS-CoV-2. So again, comparing the 2 different vaccines, so in each case, we have the red dots representing the unvaccinated controls and then 2 different vaccines. And what we can see that across these groups of 8 animals are very dramatic reductions in terms of the amount of virus that's being produced within the respiratory system, corresponding to about 1,000-fold reduction. And in many of these animals, we're seeing complete protection from infection. So in terms of proof of concept that our vaccine is going to be effective, this represents a major milestone that we've cleared. And this work, I should mention, this was done within our ferret model. In terms of timelines, what do we do next? How quickly can we get ourselves to the finish line? So we're going to be spending a lot of the summer in terms of producing the material under GMP conditions. So that's both the antigen as well as the adjuvant, so that can go into human beings. We're going to be doing more safety and toxicity work within animals over the summer, and that the hope is within the fall, hopefully by October, we're going to be able to initiate our phase 1 in combination with phase 2 clinical trials. What we're hoping for is that by spring 2021, that we could have permission to release this for emergency authorization. And hopefully by next summer, this is something that could be available to the general public. So from start to finish, we're looking at a very tight timeline. And I appreciate, to the general public, where it seems like this has been going on forever, to look at developing a vaccine over the course of 18 months might seem quite slow. But in terms of the normal timelines of developing a vaccine, this is absolutely fantastic. So it's really been amazing to see how quickly all of this has come together. One of the other things this has really done for our organization, I think it's really going to represent a defining moment and a tipping point. That one of the things we appreciated as this pandemic was occurring was really to make us appreciate how fortunate we were in terms of the infrastructure we have, in terms of the expertise we have, for our ability to create vaccines to respond to these emerging diseases, to be able to respond to these pandemic situations. And unfortunately, when you look at the various factors that anticipate pandemic outbreaks and emerging diseases outbreaks, this is not likely going to be the last one we're going to see for a while. That when we look at increased populations, the disruption of animal environments, greater human-to-animal contact, we can't think that once we're through COVID-19 that we're not going to face other pandemics. So one of the things we really want to do is to position ourselves as Canada's pandemic center. So to be able to use all of this infrastructure, this expertise, to be able to enable greater and more effective responses to these emerging diseases. And one of the things for everyone who's been trying to develop a vaccine for this, as proud as we are of what we've done and how quickly we've been able to do it, obviously that wasn't fast enough. And there's an appreciation within vaccinology that traditional approaches for vaccine development, that if you wait for the disease to emerge and start to spread, if that's when you're starting to develop your vaccine, you've essentially already lost the race, that you need something that can be applied much sooner. So what we and other groups are exploring is whole new paradigms of vaccine development, of ways to be able to develop vaccines against pathogens that don't even exist yet. So I think it's really going to be a revolutionary period in terms of what we can do and how we can apply vaccines. This represents our COVID-19 team. So we've had a large number of people who have been working extremely hard over the past 6 months to make all of this come together. In particular, Daryl, shown at the top there, has been leading these efforts to develop the COVID vaccine. But again, at VETO, we have very much a team approach to science. So this is everybody coming together to enable this type of research. There's Daryl there. And acknowledgements, the Government of Canada has been absolutely fantastic in this, in terms of providing us the resources we need so we can focus on the science. So giving us the money to develop this vaccine manufacturing facility, to give us the resources required to be able to take our vaccine through clinical trials. The Government of Saskatchewan has also been extremely generous, making sure we have the support we need, funding from CHR as well as a number of private funders. And I will stop there.
Scott, this is really amazing. From my Canadian part, I'm really, really proud. My wife just took a peek in here. She's from Ottawa originally.
Oh.
She's really excited about this. So we'll get back to you at the end. Thank you very much. Now we're going to have Sebastian. Sebastian now comes from Spain, and he's, let's look at his solution. It's really interesting. people need to go back to work, and it seems like they're providing some help with that. Looking forward to see your presentation, Sebastian.
Thank you very much, Claudio, for the introduction. It's a pleasure to be here and share this screen with Brazil and with all the countries. I think that the technology and the digitalization allows us, being in Germany, in Israel, even in Germany, in other places, to share our knowledge and our experience. So it's great that I will show you our solution that we have been working the last months. So could you see my screen? I hope, yes.
Yes, we
can. Yes, we can see it.
Perfect. I am a surgeon and a co-founder of Supermain. We are a startup focused on the development of digital health. So actually, I think that everybody knows the problem about SARS-CoV-2, and we think that there are two big issues. One is that actually there is a correct treatment, and the second is that the virus during the first week is it's asymptomatic. That's one problem. The second is that in all the countries and in all the places there are some stakeholders. One is that some are private, another are public, and they don't share the information. And actually, it's really important to have real information, and we have to share the information. And finally, the lab test. Actually, we have a PCR, a serological test, and with this great startup from Israel, this with nasal test, another one. It's really, really interesting. And the interpretation is sometimes is quite difficult. I put this tweet, it's in Spanish, but what it says is that a person that was flying from Spain, main England to an Iceland, and he took the airplane and he didn't wait until the result was done. And the problem was that he flight on a Friday and he arrived on a Saturday and he was positive. So all the people that was in the airplane must be in quarantine. So that is a big issue. In the other scenario is that each lab test has different specificity and different sensitivity. What does it mean? In this case, I will use an example for PCR. In this case, I use just two people. One is in USA and another one is in New Zealand. Where we will take the example today. Both people will have the same symptoms. In this case, in USA, we'll have fever, cough, and headache. The result is a PCR positive, and the interpretation that normally you would say is that it's a true positive. In the other case is that you have this man that the test is today. He has also fever, cough, and headache, and the PCR is sorry. In this case, is negative. So it's positive. Sorry. In this case, so you could interpret that this is a false positive. So the interpretation is really, really important because it depends on epidemiological settings. So actually, there are working in mobility credentials or immunological credentials, has different names. And first, you have a laboratory when you fill with the name of the patient and the information, and it's really easy to use. And then the patient, in this case, in the second step, the patient could have this information in their mobile phone using any kind of wallet. And finally, any kind of institution, private, public, airplane, or any kind, could check this information. So with this issue, we have developed a smart passport. What does it mean, a smart passport? It's a decentralized application that you could use in any place. It could be in Europe, in America, in this case, in Brazil. You could use, in this case, public and private. You could use in in these ambulatory laboratories, or in any case, if for going to a stadium, you could use all the information, and you will record all the information using blockchain technology. What does it mean? That all the information will be invulnerable and will be anonymized. And finally, you will have this code that is a signal code that will show you that if it's red and green is the most important, that will allow you or not allow you to pass. So how does it work? It's really easy. A patient gets in the application, he gives some, will answer some questions, some epidemiological questions, and some questions about the symptoms. Then he has the proof that could see any kind of test that I tell you, that could be a PCR test or serological test. And finally, the results are protected using blockchain. So all this process, it's really simple. The patient gets all the information, the specialist, the physician, has the test and if this test is positive or negative. And finally, due that this application is based on blockchain, it could be used in any place. I mean, you could go to a supermarket, you could go in an airplane, or you could use it in an office to return to work. What does it mean? It gives more security to all the people that is working in the office. In the other case, we gain the confidence of the people. What does it mean? If you have a restaurant and you have done this proof in the first day of the month, and you know that all the people that is working in that restaurant has a result that is negative, you get more confidence in this restaurant. So this happens in all the places, including hospitals, because in hospitals you divide the hospital in clean zones and not clean zones. So it's something similar to decrease the risk of COVID spreading. Actually, we have been working in the authorization of the informed consent of any kind of medical surgical treatment during the last years. During the pandemic, we begin to work in a smart passport. And what is really is another point is that this interpretation of the results, we are working in our first minimum product viable using artificial intelligence to get faster to the results. And this, we hope that in the next months we will get our tool in a lab pilot, and it could be really useful because it is really is scalable. What does it mean? That we could use in Spain, we could use in other countries, and due to the technology that we use, we could share, and it's anonymized. So that is a good point for everybody, because we respect the rights of everyone. And finally, this is the team the founder team. We are a group of surgeons that are dedicated in the digitalization of medical process. So this is our project. I hope that you could understand what are we doing and what we expect in the next weeks. Thank you very much.
Thank you very much, Sebastian. I think this is a very ambitious and very interesting project. I think, hopefully in the coming weeks or months, we're going to see some of that. We're seeing a lot of demands for people to show their passports, do PCR before traveling. I think the world needs to be traveling again, to feel safe. Companies, it's a real challenge. By the time the guys that work at vaccines are working really hard, but it's still have a long ways to go to have everyone immune to this, or some kind of immunity at least. So congratulations on the great work. So coming now to the last presentation, then again, we're going to have questions and answers. We're going to have now Mustafa Guteokin from Inovaco, also in Spain, right? Hola, ¿qué tal? Muy bien. So again, you know, be on the softer side, but with a different perspective.
Let's share my screen. Can you see my screen now?
Not yet.
Okay. Can you see right now?
Yes.
Okay, perfect. This is better, I think. Hello, everyone. Thank you for being here today. My name is Mustafa. I'm a research and development engineer at Inovaco. Today, I'm going to explain our artificial intelligence solutions on COVID-19 detection, and I will also talk about the problem we observed, and I will also talk about our approach and our solution. But before that, I would like to talk about our company. We are Industry 4.0 system integrator that using high technology like artificial intelligence, augmented virtual reality, and machine vision. Our team consists of 7 engineers. We have been established in January 2018. Where we have 2 offices. One of the offices is in Vitoria-Gasteiz in Basque Country in Spain. The other offices in Istanbul at Turkey. Our company also have been supported by well-known organizations and corporations from both Basque Country in Spain and also from Turkey. As I mentioned,
Mustafa, sorry for interruption. This is Emin speaking from Inovaco. Mustafa, on the right-hand side, you have a window which is closing the screen, so we cannot see it. Could you please remove it? That one, yeah.
Sorry. Can you see it right now?
You should no, now it's in the bottom.
Sorry. Is it okay for now?
Now it's fine.
Okay, thank you. As I mentioned, we are a high technology company and our core activity area is factories. We use artificial intelligence and computer vision as main part of our technology. And we usually use this kind of technologies for surface inspection. I would like to give an example to figure out that how we are doing surface inspection. Let me give an example from the car industry. During the car manufacturing, usually there are some operators that making surface inspection on the car surface, but we are making this process automatically with using deep learning technology. With deep learning technology, we are making automatically this process and we can find all the defects and other problems on the car surface. Before I talk about our background of COVID-19 study, I want to talk about how did we start our journey for this project. Approximately 6 months ago, we were considering about the opportunities and challenges on using artificial intelligence and deep learning for medical areas, for the medical field. And then we started to make some research and study on how can we use medical images for the feeding deep learning models. After our research and studies, in the beginning of the COVID-19 pandemic, we decided to start our project, which name is COVID-19 detection. But one of the challenges about this project was the general detecting COVID-19 problem is that limited time. If we can decrease this time, then we can save more lives and we can help more doctors and it will be a better result. Our goal was building an artificial intelligence model that can assist the radiologists for better and faster inspection. Our solution for this approach was very simple. We are feeding artificial intelligence model with X-ray images, and we are trying to make a good classification and detection. Also, I want to mention that why we use X-ray images, because there are some approach that people can use CT images for detecting COVID-19 for this kind of projects. The reason is that we use X-ray images because when you go to doctor or when you go to hospital, firstly they are taking your X-ray image, and if we can detect and if we can find the COVID-19 in the first step, then we can be more productive and we can be more effective on this approach. We can save lots of lives. We believe that. That's why we choose X-ray images on our project. We believe that we made really significant progress on this project. We have really good accuracy on detecting COVID-19. We can classify the people who has COVID-19 and healthy people. We are actually saying that to our model, if let's say this is the image coming from the people who has COVID-19, and this is the image coming from the people who has not COVID-19, and if our model can understand the relation between these images, and if it can understand the relation between these images and patterns, then our model can understand the problem and generate a solution for us. But of course, we can expand our deep learning model for next studies. We are still currently working on that. What can we do more for this project? As I mentioned, we can add some more lung disease for our model, and we can expand the capability of our deep learning model, and we can convert our approach in a trustable industrial solution for hospitals or for medical doctors. But of course, we need some support, we need some cooperation, we need some consultancy and collaborations. If we can have cooperation with healthcare establishments like clinics, hospital, or labs, we can get better data, we can get more data and feed our model with the better data, and we can get good results from our model, and we can increase the accuracy of our model. We can also make a consultancy with radiologists, and we can make collaboration with academics to interpret this kind of data, because you know that as an engineer, it's really hard to interpret medical images because we don't know where is the exact problem, where is the problematic part of the image. But if we can make a consultancy and collaboration with the experts, they can help us to understand what is the problem, and then we can make a better approach for generating solution. I want to show some tested images from our model, but firstly, thanks to Johns Hopkins University, from the beginning of the pandemic, they share all the images from the patients, and they gave these images for the making a good technology to solve COVID-19 detection problem. And we use this kind of images for feeding our model. But unfortunately, this just using directly these images not enough to have successful model. You need to make some process on these images, like data augmentation, image pre-processing, because you need to increase amount of data and you need to extract features better than the normal images. That's why we use this kind of techniques to improve our results. We increase the amount of data and we increase the quality of data. And we got these results. As you can see, there are 3 people who has not COVID-19, and there's a people who has COVID-19. You can see the accuracy in here. In conclusion, I tried to explain some of the challenges about the COVID-19 detection, and I tried to explain our approach. We believe that we can improve our results and test images, but as I mentioned, we need some more support and collaboration with the experts to improve our results. In the end, I also want to say thank you to Albert Einstein Hospital, EBN, Enrich, Eretz for this event and inviting us. I want to also say thank you to Big Arabia for all their support. If you have any question, I would like to answer them. By the way, this is our contact informations. If you have any question or talking about something, you can contact me directly from my email or my telephone number. Thank you again.
Thank you very much, Mustafa. So we've come to the end of the presentations. As you could all see, these are amazing people working on the cutting edge of new technologies, trying to get us all out of this pandemic situation with different tools and solutions and so forth. So, you know, we're here, of course, it could be anywhere. I mean, there could be people from in the moon watching us now, but in fact, most of the audience is from Brazil. So start, you know, this Q&A session with one general question for all of you, if you can, starting, you know, with you, Mustafa, and then we could like walk back on this, which is, you know, you came here, you presented, and you know, all of you. So, what would be, you know, ideal outcome out of this presentation? At least with, you know, Brazil or Brazilian companies. So what do you get out of this that to make you happy? So can, you know, everyone has a boss. I have a boss. Everyone has a boss. Maybe, you know, founders don't have a boss, but, you know, what do you like to get out of this presentation, the time you spend here, other than learning about these amazing people and technologies? So we can all turn on our videos now. So be interactive, so it's more interesting. Thank you. So, like, who'd like to start? I mean, I can call people, but you know, what do you like to get out of this? You know, be useful for your company and for the relationship that you're starting to establish here.
I can start if you'd like.
Okay, let's go.
So I think as far as NanoScent goes, we would really like to have our device, our technology. We need to get to the final step with our technology, and that includes having clinical trials, validation trials, just in order to really put the device out there.
Great. Anyone else?
Well, I would like to take the word for a couple of
yes.
seconds. By the way, this is Emin from Inovaco. I'm the founder manager. I'm a colleague of Mustafa. So, well, it was really interesting, and I was really impressed to see all those other startups and other institutions here. Really, we are really proud that we are one of those selected organizations. And congratulations to everyone. As I mentioned, I was really impressed. And as Mustafa mentioned, from Inovaco perspective, it would be very interesting for us to continue our project somehow with new data. So if we can fit our deep learning model with new data coming from labs or coming from hospitals, coming from institutions about health, we are totally confident that we can create a ready-to-use artificial intelligence model to inspect X-ray images. So we are extremely looking forward for this opportunity, and I believe we are at the correct point to do it.
Great. Next volunteer, before I choose.
I think for us, I mean, I think there's two opportunities. I think on the one hand, we're interested in clinical trials for monitoring of Alzheimer's disease, again, because we can measure this progression, but also we're really like launching in the market. We have the ANVISA approval. The technology works great. And I think for like screening, so it's not a standalone diagnostic, it's a companion diagnostic for a doctor to feel more comfortable and more confident also to make an early diagnosis. So I think like memory clinic, kind of geriatric screening would be the opportunity for us in this context. And not necessarily only for people after like the age of 65, for example, when Alzheimer's officially starts, but we can start as early as 55, but it's also important to create a healthy baseline as well. So even before that, it's important for you to know what's the healthy baseline for you to then compare, okay, when are things starting to go wrong and decline? So we can really start as early as 40, 45 years old.
Very good. So, Sebastian or Scott?
Okay, I will say that this is an amazing opportunity for all the startups, and it's really interesting that we are in several places around the world with different technologies, with different experience, and with different milestones to accomplish. What does it means that are really, really beautiful projects that I have seen. One has different aspects to improve. One maybe, for example, is more related to people that has more age, another that need more data, and are some issues that each of our startup must win. In our case, I think that the most important is this kind of networking, this opportunity to share our knowledge. I mean, this is the previews of a really interesting conference, and we hope or we think that the digitalization in the healthcare sector has arrived. Some technology has been for a long time. What does it mean? That artificial intelligence, face recognize or another technologies has been for 10 years, maybe more, and we have not used it due to a problem that our colleague has said, the data. The data is, all the hospitals, all the institutions are really jealous. I don't know what else to say. And I hope that, I hope that COVID has changed for a benefit of the humankind. What does it mean? That all the data, we are trying to improve the healthcare sector independing of the that you live, independing if you are public, private, it doesn't matter. The most important is to work together in this, how would I say, electronical, digital networking, and try to connect and do more interesting things.
Very good. Thank you very much, Sebastian. You know, very great hopes, and I share some of the same hopes. Scott, I think, you know, you have some words to say as well.
Yeah, I agree 100%. This has been very inspiring, that this has been a pretty dark time for all of us. To me, one thing that's very clear, there's not going to be one magic bullet that's going to get us out of this mess. In particular, in the early stages of getting this under control, that we're going to need these different approaches in terms of controlling the disease, in terms of identifying infected people, in terms of understanding the pathologies associated with it. So it's wonderful to see so many people coming at this from so many different perspectives. One of the things that we could certainly offer to that in terms of animal models of disease, where you have the opportunity for more controlled infections, the opportunity to be able to get samples, is certainly something we would be interested in working with people with. But then also in terms of for us to be able to evaluate the efficacy and safety of our treatments. Some of the imaging approaches I saw here, some of the detection approaches, I think would add a very nice component to our efforts. So very exciting stuff.
It's great, Scott. I think, you know, this is the other thing, you know, of course, I mean, you have the Brazilian audience here, and myself representing a large healthcare institution. Certainly we'll follow up with each of you to see if there's any collaboration that you could have with our own research institute and innovation and incubator as well. However, I mean, you know, you all met each other, you know, as Scott just highlighted. And so hopefully, you know, these solutions cross, you know, across other countries as well. So I think, very close to the end. So I'd like to say thank you all. This is a very different way of doing conferences. I kind of like that because, I just walk with my dog in the morning. Now I'm here, then, have lunch very quickly. So I guess it's changing for everyone, right? So hopefully some benefits coming out of this. On behalf of Hospital Israelita Albert Einstein, thank you very much. Your time is very precious, and hopefully it'll be good for all of us, things like that. So I say thank you. If anyone want to say 2 last sentences, please do so, and then we say goodbye to the audience. That's all. That's it. Ciao. Goodbye.
Ciao. Goodbye. Bye-bye. Bye-bye.
So we can all learn a word. Ciao.
Bye-bye. A pleasure.
Bye-bye. Have a great day, all of you.
The same for you. Have a great day.
Thank you, you too.